Okay, I have 3 o'clock Eastern time here, so I'll go ahead and get started for the webinar session today. Welcome to the Collaborative Science for Estuary series. This is Nick Sobral with the Science Collaborative. Today's session features a collaborative research project that used passive acoustic monitoring to evaluate how fishery species use the Hudson River Reserve in New York. We have some folks on the project team joining us today to share more about that work, but as always, we have a quick introduction to the reserve system, the Science Collaborative, and this series before we get started with the presentation. So the National Estuarine Research Reserve System is a national network of 30 unique reserves as shown on this map. It's a NOAA program that works in collaboration with a local place-based partner, either a university, state agency, or nonprofit at each site. Each reserve site includes programs focused on the activities you can see on the slide, which include land stewardship, research and scientific monitoring, training, programs for the public and local officials, and education. The Science Collaborative supports science for estuarine and coastal decision makers by coordinating regular funding opportunities and supporting user-driven collaborative research and transfer activities that address critical coastal management needs identified by the reserves. The Collaborative Science for Estuaries series features project teams supported by the Science Collaborative, program staff, and partners. Speakers share their unique approaches to addressing current coastal and estuarine management issues. A quick note on informed consent. Webinars are a valuable learning opportunity for us as a program. Insights from speakers and participants like you can help inform how we support others in doing collaborative science, for example, in the development of future workshops, webinars, or resources such as tools, tips, and case studies to add to our guide to collaborative science, which is open access. With that in mind, we just want to inform attendees that any insights or feedback you share, for example, in the chat or via the exit survey at the end, may be used for program learning and development purposes. Please note that if we capture a direct quote from you that we'd like to use for learning purposes, we will reach out to you beforehand to seek permission and provide an explanation of its use. All such quotes would be used anonymously only, and we're grateful for the opportunity to learn alongside all of you. A few quick notes on housekeeping. All attendees are muted upon entry. We'll be handling presentation-related questions via Zoom's Q&A feature. You can enter questions for presenters as they occur to you throughout the webinar, and we'll plan to address as many as we can during the Q&A portion at the end. You'll also be able to come off mute and verbalize your questions if you choose to do so by raising your hand, and organizers can see it and will confirm with you ahead of time that you're ready to come off mute and ask your question when it's your turn. Chat is visible to everyone, so please use it to talk amongst yourselves. You can also use it to ask organizers any questions or let us know if you're having any technical issues. And one last note, later in the presentation, there are some audio files that would be better if you have headphones to hear, so I recommend that you put headphones in if you have them. And with that, I will introduce our speakers for today. Joining us today are Maya Niemisto, an environmental science specialist with New York State Department of Environmental Conservation and the Hudson River National Estuarine Research Reserve, and Rebecca Cohen, a postdoctoral fellow at the K. Lisa Yang Center for Conservation Bioacoustics at Cornell University. We also have some folks joining us for the Q&A, so they're just in listening mode right now, but Aaron Rice, who's the PI for the project, Patrick Baker, and Chris Bowser, so you might hear from them later on during the Q&A portion as well. And with that, I will pass it over to Rebecca to take over.
Great, thanks. All right.
All right. Hello, everyone. Thank you so much for that wonderful introduction. My name is Maya Niemisto, and I am calling today in from the Hudson River National Estuarine Research Reserve. And I'm going to kick off this talk about aquatic acoustics as we are listening to fish in the Hudson River National Estuarine Research Reserve in New York. You can see listed our collaborative team and all of our respective affiliations. And we're so excited today to be talking to you about a number of different components to this project. And we'll walk you through some of our findings, some of our outputs, and share some of the exciting parts about this research collaborative project that we've had going on. So first of all, I wanted to highlight some of the Hudson River National Estuarine Research Reserve needs that were kind of put forth by the collaborative team and how our project has moved towards trying to address these needs. So one of the kind of key stated needs early in the kind of project development was a need to understand how some key fish species use the Hudson River and the Herner component sites as habitats. We were able to address this, and we'll kind of walk you through some of these findings. We were also looking at monitoring some protected species by using a cost-effective method that will also continually minimize harm to some of these key species. So that don't incur any kind of physical stress or elicit some kind of behavioral response. Most traditional fisheries methods involve catching the fish, which does cause some physical stress, or could elicit avoidance behavior where fish will try and leave the area if they know they're being netted. The methods that we'll show you today using these passive acoustic monitoring technologies do not cause physical stress or behavioral response in fish, so we're able to better understand how those individuals are behaving below the surfaces of the Hudson River waters. We also had the opportunity to utilize some of the Hudson River National Estuarine Research Reserve water quality and meteorological monitoring data sets, including the CDMO data that helped us understand some of the ecological interactions that we were seeing with the acoustics. And at least for me, one of the more exciting kind of outputs that we were able to develop was some of this continually fostering public engagement and stewardship. So using sounds in order to reach new audiences and develop some new educational tools. So this is just an overview of what you can expect for the next hour while you come along on this ride with us as we listen to the sounds of the Hudson. We'll share some of our key findings, some of the challenges and opportunities that presented themselves throughout this project, some of the really kind of organically grown follow-on projects that we're very excited about. The Science Collaborative Research program in so many ways was very successful with our team in that our team was built and we have a lot of follow-on enthusiasm and projects in the making. We'll share some of the specific outputs that we have, including some of our educational products that are now available, both online and at the headquarters of the Hudson River National Estuarine Research Reserve at Norrie Point Environmental Center. And then we are going to share some of the lessons learned and some of the future directions that are kind of growing from this project in order to share things that may be of use to others who are interested in or potentially pursuing similar research in their reserves.
All right, so with that, I'm going to take over. And I'm going to walk you through some of our methodology and some of our findings. And I'm going to start out with a little introduction to passive acoustic monitoring, because I recognize that it's not a methodology that is necessarily very familiar to a lot of people. At its most basic, passive acoustic monitoring simply entails using sensors to passively record underwater or terrestrial soundscapes in order to capture signals of interest, which may arise from biological activity, from natural phenomena such as rain and weather events, or from human activity. And the schematic on the bottom here walks you through kind of the workflow for typical to passive acoustic monitoring. As we record and digitize sound, then there are two avenues that are commonly used for analysis. One is kind of a high-level holistic approach, which looks at the soundscape by looking at the amplitude of sound in different frequency bands. And another is more of a species-specific approach. And this typically identifies vocalizations or calls of the species of interest in order to construct time series of acoustic activity and an animal presence. This approach can also be used for constructing time series of presence of non-biologic sounds, such as human activity and other natural phenomena as well. That's the kind of how of passive acoustic monitoring. Maya mentioned some of the advantages of it, such that it is completely non-invasive and unobtrusive, which is really advantageous when you're working with some of these sensitive species, especially endangered species, for which it's really not optimal to be capturing and handling animals consistently. So in addition to being non-invasive and unobtrusive, you can also get really fantastic spatial and temporal kind of coverage of your data. We did continuous recording at most of our sites, which means that we were recording nonstop throughout most of the spring, summer, and fall. And by recording at a number of sites spread across the river, we covered quite a bit of the ground. You might be wondering, though, why passive acoustic monitoring was our first choice here. Well, in addition to the advantages that I listed, another is that work over recent years, and actually extending back several decades, but a lot of it in the last decade or so, has revealed that a lot of fish species actually produce sound for various life history needs. More than, well, about 1,000 fish species have been found to actively produce sound. And that's actually about 83% of species that have been evaluated for sound production. So it seems like a pretty high rate. This is aligned with some phylogenetic work, which seems to suggest that sound production is probably an ancestral trait to most ray-finned fishes. And it's arisen multiple times in the evolution of the 35,000 or so ray-finned fish that are currently extant. Okay, so into our methodology for this project. I mentioned that we were recording across a large span of the river. What you're looking at here in the map on the right are the 5 sites that we were recording at, which include the 4 Herner component sites, that's Piermont, Iona, Tivoli, and Stockport, as well as a site right at the Norrie Point Environmental Center near Hyde Park, New York. And our approach to data collection was kind of a 2-pronged approach. We deployed devices both in the middle of the river channel, to our main river channel habitat, as well as in some tributaries and wetlands adjacent to the main channel. So in the river channel habitat, we deployed sound traps, and these were sitting down on the bottom of the river and recording. While in the tributaries and wetlands, we used Swift units, which had been custom modified to take cabled hydrophone input. So we actually just would strap a Swift to a tree and then run a cabled hydrophone out into the water adjacent to it. And we collected data in the spring, summer, and fall of 2023 and 2024. And in total over that time, we recorded about 2,700 days of cumulative acoustic data. So as I mentioned before, it's pretty easy with passive acoustics to obtain these really large data sets, really rich with great temporal coverage, and in this case, great spatial coverage. This spanned about 100 miles of river, if I didn't mention that already, which is pretty neat, a really cool latitudinal gradient and salinity gradient. And now the challenge is just analyzing all that data. So I'll go a little bit into some of the approaches that we took for analyzing. First off, one of the most common things to do is just to visualize your sound data and look at broad patterns across your sites or at each site through time. So what I'm showing you here are called long-term spectral averages. These are a common visualization of sound data, which presents frequency on the x-axis, whoops, little spoiler there, frequency on the x-axis, time on the y-axis, and then the amplitude of the sound, or how loud it is given by the color. So in this case, hotter colors show where the higher amplitudes are. And we're looking at 3 weeks of data across our 5 main channel sites here. And you can see that right off the bat, there's some really striking differences in soundscapes between these sites. Most of what you're seeing here is due to the differential presence of vessel noise, of boat noise at these different sites. You can see at Tivoli and at Hyde Park especially, there seems to be a really clear day-night signal. So actually, let me see. Can you all see my cursor right now? Yeah, great. So we see these patterns of high amplitude, low amplitude, high amplitude, low amplitude. Those are daytime, nighttime, daytime, nighttime. And that reflects the activity of both commercial and recreational vessels at both of these sites. The Hyde Park recording location is right next to a marina, or a yacht club rather, the Poughkeepsie Yacht Club. So that's an area that has a lot of small vessel activity. At Iona, there seems to be a lot of vessel activity as well, but it's not as neatly partitioned, I'd say, into those day-night cycles, which is kind of interesting. Way up at Stockport, that's a much quieter and narrower and shallower part of the river, and that's reflected in the lower levels of anthropogenic usage. And then surprisingly, Piermont is kind of similar to Stockport in these lower levels of vessel noise, which was something that we didn't expect. And it may also to some extent reflect just where we put our device in the main river channel. Perhaps we just were not in a good position to be recording some of the vessel passages down there. If we kind of summarize these plots just to look at amplitude versus frequency averaged through time, that would look something like this. Now we're looking at spectral probability density plots. So on the left, on the y-axis, now we have amplitude. On the x-axis, we have frequency. And the color gives you the probability that that frequency will be at that amplitude. So where we have red, that's saying that most of the time at Hyde Park, what frequency are we at here? Let's go here to 100 hertz. Most of the time, 100 hertz at Hyde Park is about 60 decibels. Most of the time, 1,000 hertz at Hyde Park is more like 75 decibels or so. But the takeaways from this figure are just that both Stockport and Piermont, we can see that, you know, above about 100 hertz, most of the time they're very quiet. The red is down in the lower end of the decibel levels that are recorded there, whereas at Hyde Park and to a lesser extent at Iona and Tivoli, it's more likely to be loud in these higher frequency bands, and that's due to the contributions of vessel noise. So we're experiencing very different soundscape conditions across these sites, and of course this has implications for things like acoustic communication and perhaps also behavioral response to either vessel noise or like the physical presence of a vessel. That's all I'm going to go into for soundscapes. We're going to turn right now into the species level analysis because that's what we mostly focused on for this. And we're going to start with Atlantic sturgeon. So Atlantic sturgeon were one of the first species that we wanted to target with this project. They're an endangered species. They used to be really, really abundant all along the North American East Coast. They're an anadromous species, so the adults spend most of their life out in the marine environment, but they return to their natal rivers for spawning on an interval of a couple to several years, depending on if they're male or female. And so when they come into these rivers for spawning, they were targeted very intensely by fisheries, all from the colonial times right through the early 1900s, at which point the fisheries really collapsed. You know, they're these really interesting, long-lived individuals, very low reproductive rates, et cetera, and they just couldn't keep up with the fishing pressure. And while there's been a moratorium on fishing of Atlantic sturgeon for decades now, their populations really haven't rebounded. And there's a lot of data gaps, so we just don't understand what might be inhibiting their recovery. So they're a high priority for the New York Department of Environmental Conservation Fisheries Unit to be investigating and figuring out better ways to manage and help them to recover, hopefully. Sound production has not been reported for Atlantic sturgeon, but it has been reported for a number of other sturgeon species, most notably lake sturgeon, which are similar except that they are not anadromous. But lake sturgeon produce these low-frequency, thundery sounds when they're spawning, and this has been used to it's kind of diagnostic of their spawning activity. It just so happens that the largest remaining Atlantic sturgeon spawning aggregation occurs each year in the Hudson River. And so with this lake sturgeon spawning queue in mind, we went ahead and put a recorder in the Hudson River right at this Atlantic sturgeon spawning aggregation. And we recorded during the spawning season. And we were pretty excited when we looked at the data because right off the bat, we found these low frequency thundery sounds that looked and sounded a lot like lake sturgeon spawning sounds. When we kind of counted up the occurrence of these putative Atlantic sturgeon thunders, we found that the occurrence of the thunders was really tightly correlated with the occurrence of telemetry tagged fish. And if you want to hear what these thunders sound like, let me see if it'll play for you. I hope you were able to hear that. That was two different thunders. They're kind of just a low timpani roll, quite low frequency though, and rather low amplitude. While we were excited about this correlation between thunder occurrence and telemetry tagged fish, that obviously is not like a definitive attribution of sound to species. So last summer in 2024, we got an opportunity to go make some recordings of captive spawning Atlantic sturgeon down at the Bears Bluff National Fish Hatchery in South Carolina. And these animals in the hatchery spawn spontaneously each year. They don't introduce hormones in order to induce spawning, but they regulate water temperature and photoperiod, and then they naturally spawn each year. And they've been doing so since 2011, which is a really unique condition for captive spawning of fish. Usually hormones are used to induce spawning. And so we had high hopes that these animals would be exhibiting naturalistic behaviors in the spawning tank. So last summer we brought a recorder down and we recorded in the tank for about a month, all of August. And during that month, the fish in our tank spawned one time on one day. Actually, sorry, it was multiple spawning events over the course of one day. And on that one day, lo and behold, what did we record but more thunders? So now I'm showing you a few different visualizations of sound here. All of the plots on the left here were recorded in the Hudson River. Here's a representative example of one single thunder. This is a spectrogram similar to the LTSAs you saw earlier. So again, frequency on the Y and time on the X. Here's an acoustic waveform that corresponds to the spectrogram. Same things for a representative thunder recorded in the hatchery, the spectrogram and the waveform. And down here, I've averaged the power spectra of a number of thunders from the river versus from the hatchery, as well as some background noise samples. And so those show, again, amplitude versus frequency. And you can see that the noise conditions in the tank setting were not as wonderful as in the river. But nevertheless, we recorded multiple instances of this same thundery sound, and they only occurred on the days, that one day where spawning occurred in the tank. So this was a really exciting finding for us, in addition to being just new insights into the behavior of these animals during spawning activities. It has immediate utility for monitoring of these populations in all of their spawning rivers. We could use this to look at things like the phenology of spawning, getting some either relative or perhaps even absolute estimates of aggregation size at these spawning grounds. And this can also be really great for identifying spawning sites that are not currently being protected or studied, rather. And this is in press right now in endangered species research. So hopefully this will be out within the next few weeks. Now, you may have noticed in that LTSA that I showed you with the sturgeon thunder, whoops, getting stuck, that there was another dominant signal present here, namely this kind of continuous band of energy between 200 and 400 hertz. And we were able to identify that pretty quickly as chorusing freshwater drum. So when male freshwater drum are ready to mate, they advertise for mates by calling. And it's usually from late afternoon through the middle of the night or so every day during the spawning period. And when multiple individuals are calling repeatedly over and over and the calls all overlap, you end up with this kind of continuous drone of energy in the frequency band of their calls. That's what we're seeing here. Freshwater drum are non-native in the Hudson River. They've been there for a few decades, but they really haven't been studied in the Hudson River. And so the extent of their colonization and their interactions with native species are not at all characterized. We don't really quite know what they're doing there. And actually, before I move on with that, I wanted to play you a little bit of sound if you can hear this. This is an example of what individual freshwater drums sound like. And you can hear a few different individuals. But when they really get going, and there's a lot of individuals calling simultaneously, it sounds like this. So kind of neat, pretty loud. When we looked at those long-term spectral averages, those LTSAs, which were visualizations of long periods of sound at our sites, what we noticed is that at the Atlantic sturgeon spawning site, the Hyde Park site, freshwater drum chorusing really dominates the soundscape every day during the same spawning season as the Atlantic sturgeon are there. So on this LTSA, you can see these little hot clouds of acoustic energy spanning about 200 to 400 hertz every day. This is 1 week of data. You can see that there's a daily chorus, and each day this chorus lasts for hours. It goes from like 4:30 until midnight or so. When we looked across sites, we saw that the Hyde Park site really seems to be a favorite of the freshwater drum. Choruses are present, but lower in amplitude and for less of the season at Tivoli and at Iona. Down here at Piermont, we thought initially those might be freshwater drum, but closer investigation showed that they're more likely Atlantic croaker. But it was pretty interesting to find that the freshwater drum were so predominant at Hyde Park. Our New York DEC fisheries collaborators weren't aware that they were so heavily using this same space. And so that raised some questions of possible competition with the Atlantic sturgeon, either for actual physical spawning grounds and spawning space or for other resources at that site. And so that led to one of these follow-on projects that I'll go into a little bit later. I'd like to address some of the kind of challenges and opportunities that came up during this project. Bioacoustics is a really popular methodology for marine species, and it's been pretty well developed there. And there's a lot of species which have been acoustically characterized. Once you've got an acoustic characterization and you have one or more signals that you can reliably attribute to a particular species, using acoustic data to infer the presence of that species becomes pretty straightforward, and it becomes a really powerful tool. But until you have that kind of that groundwork laid, you're a little more limited in what you can do with acoustics. So coming into this project, there had been very, very little in the way of acoustic characterization of fish species that occur in the Hudson River. So that's why we started there with, for example, the Atlantic sturgeon. Another species that we wanted to look into were short-nosed sturgeon. Now, these are not in the same endangered state as the Atlantic sturgeon, but they're still a protected species. Recent work suggests that the population is actually rebounding quite well. But again, being able to use a non-invasive and unobtrusive approach to continuing to monitor the species would be a boon to the fisheries unit. So we went ahead and we made some recordings at the site of the short-nosed sturgeon overwintering aggregation, which is near Esopus, New York. And at this site, tens of thousands of individuals come and aggregate and just spend a couple months in kind of a very low activity state while the water is very cold. And during that time, we recorded some really weird and funky sounds, and I'll play a couple. They tend to have this like plinky characteristic to them. And I'll play a few for you. Here's one. Actually, that one I call chittering. Here's another. And then when I go to the next one, I imagine it's going to play all three of these again. There we go. And that one is still going. So we recorded these again at a site where there are tens of thousands of short-nosed sturgeon present. There are probably some other species present as well. We expect them to be not very active at this time of the year, but we can't yet rule out that these sounds came from another species. So this is something where we need further validation. That's hard to do, though, in this river environment where the animals are deep and the water is turbid. So what we're working on right now is trying to identify other data streams that could be complementary to this acoustic data and allow us to get a better sense of exactly where animals are relative to the receiver, exactly which animals or which species are present, etc. At one of our other sites, though, one of our tributary sites, we didn't have as much of a problem with depths and turbidity. So in Black Creek, which is kind of right across the river from Norrie Point, right near the site of our recorder, which is actually, you might be able to see the rebar sticking up out of, there it is, the rebar is sticking up out of the water right there that our recorder was attached to. At this site, the water is only about, 4 to 5 feet deep at high tide, and it is crystal clear oftentimes, which is really incredible. And so this past year, what we piloted was an approach of putting out a time-lapse camera right co-located with our recorder. And we were able to get some really nice and clear images of fish species. And what we're working on right now is going through and identifying the species in these images. The first pass of analysis worked on comparing the acoustic diversity index with Shannon diversity as calculated from the fish images. But our kind of next steps in this project will be then to compare presence of, or rather to identify sounds which seem to be correlated with the presence of each species. And so hopefully this can help us get at some of those really useful species-specific signals that we could then use for monitoring these species beyond Black Creek. Aside from sound characterization, another challenge that we hit up against was kind of more logistical in nature. So in Black Creek, we had hoped to make use of data which had been collected in previous years by a weir. So a weir had been deployed in previous years to funnel fish through one single kind of passageway where they could go upstream or downstream. And that passageway was equipped with an optical counter. Now, Black Creek hosts a big spawning run of river herring every year. And so we had hoped to use the weir counts of the spawning river herring in order to get a better understanding of when the fish were moving through and how many they were, and use that to identify an acoustic signal that might be used in future to monitor the herring runs. Unfortunately, the weir was a really challenging piece of equipment to work with. It was damaged repeatedly by high flow events and washed right out of the tributary. The optical counter needed to be reset manually every day, sometimes multiple times a day. And so the fish count data that we actually were able to get our hands on was very limited and had kind of inconsistent temporal resolution. And we ended up having very little overlap between fish count data and acoustic data at this site. We went ahead and tried to work up what we had. You know, it was really just a few weeks of data of some pilot acoustic data that we had from 2021. We compared both fish counts and our acoustic data to, or rather we correlated it with some water quality and meteorological variables. But we only found very weak correlations between any of those. What you're seeing on the top there is the correlation between fish counts and those water quality and meteorologic variables. If we just look at this column on the left, you can see that most of these values are like 0.2 or less, so nothing really strong and convincing. What the bottom figure is showing are fish count, or the thick blue line, and then we computed sound amplitude levels in several different frequency bands and third octave bands actually across the period where we had overlapping data between the two. And you can just see visually, we computed the correlation and it was pretty much zero, but you can see visually that the fish counts do not seem to be correlated at all with sound levels in any frequency band. But one shortcoming of this analysis, in addition to the fact that we only had a very short record of acoustic and fish count data to work with, is that the water quality and meteorological variables that were available for this site were in fact from sensors that were on the other side of the river. So there's kind of a spatial mismatch between the water quality and meteorological variables and the acoustic data and the fish count. So there's a lot of room for further work in this. I think that there's still a lot of opportunity to identify a sound that could be used to monitor those herring runs, but we need to identify some other data streams that we might be able to complement our acoustic data with. And then one more opportunity for kind of learning that came out of this project was in terms of mooring design in a busy and high flow river environment. So the moorings that we put out in the first year of the project were designs, they were based loosely on a design that our DEC fisheries collaborators used for deploying telemetry receivers. But well, I'll walk you through that first. So it was a cinder block, which we drove a piece of rebar through, and then we strapped our recorder. This is our recorder right here. We just strapped our recorder right onto that rebar. We had a couple of U-bolts set in the cinder block as well, which we could attach line to so that we could set out an anchor. And on the other one, we attached a float so that we would be able to locate our devices more easily. And these are recovered by grappling. So you drop down a grapple and you try and catch on this anchor line, which in the first year we put out about 30 meters or about 100 feet of line, thinking that that would be an ample target to catch later on, knowing the location of both the anchor and the block. What we found in year one, though, is that brand new floats are very tempting for a lot of recreational river users. And so we had a number of floats removed from our devices, unfortunately. We also had some well-meaning Good Samaritans who pulled up a device despite the sign on the float saying, research, do not touch, Cornell, New York DEC. They pulled it up and then they were kind enough to contact us and return it to us. But unfortunately, that cut a deployment quite short. We also, for one of our sites, experienced a little bit of movement of the mooring. So despite the anchor set upstream to prevent blocks from walking, one block did walk just a little bit, and so we had to pull it out before it was potentially swept away. And so in year two, we completely skipped the surface expression, and we increased the length of this anchor line to kind of offset that. So even though we wouldn't be able to find our device as easily just by using the float, we knew that by recording the coordinates where we dropped the anchor and where we dropped the block, we would know then more or less where the anchor line was lying and have a big target to be hitting for scooping it back up. And we ended up with a 100% success rate for recovering these despite not having the surface floats on them. So that was just a nice opportunity for us to learn from some of the experience of our DEC collaborators who have been deploying devices in the river for a long time for a number of different equipment needs, and use that to improve our acoustic mooring design for this setting. All right, now I'm going to go into just a couple of the follow-on projects that arose from this. The first one has to do with invasive plants. So I touched briefly on those non-native freshwater drum, and non-native and invasive species are a high priority for DEC fisheries managers as well as the Herner reserve managers. If you've ever been to if you've ever been to the Hudson River in the summertime, you'll be familiar with the water chestnut, which is a non-native and highly invasive plant that's been growing in for the last 100 years or more, unfortunately. What you're looking at in these two photos are the very same perspective of the river. So standing on the deck at Norrie Point Environmental Center, looking southwards, and these are separated by just 6 weeks. And you can see that in those 6 weeks of the late spring and the early summer, the water chestnut just fills in these slow-moving embayments of the river and just so densely vegetates them that it looks like a solid meadow. In addition to the physical changes that come with this, water chestnut is known to create very low oxygen conditions as a floating aquatic vegetation. It is not putting much oxygen into the water, unfortunately, and so oxygen gets drawn down underneath the water chestnut by respiration and also to some extent by the water chestnut itself at night. And so what we wondered is that in addition to these physical and chemical changes to the habitat, what are the impacts that water chestnut has on the soundscape, and can those changes or can those impacts be used to measure kind of the health of communities, either in water chestnut versus outside of water chestnut, or in areas where water chestnut is going to be removed or where it's growing in, etc. So we put out recording devices around Norrie Point Environmental Center, one in the depths of the water chestnut bed, one on the edge, one in the open water beyond the water chestnut, and then a couple up here in some mostly native submerged aquatic vegetation beds as well. And we had oxygen data to go with a couple of these sites. We had an oxygen sensor that we deployed in the water chestnut bed, co-located with our recorder, and then the Herner water quality station at Norrie Point is located right here on this dock, and they do they do oxygen measurements there as well. So our first look at the chemical environment showed what we would expect to see, that the oxygen levels in the water chestnut bed, which is the green, are much, much lower than up in the submerged aquatic vegetation bed. And I thought it was also interesting to see that the amplitude of the tidal oscillations, the tidal fluctuations in oxygen, were much more pronounced in the water chestnut bed than up in the submerged aquatic vegetation bed. And of course, this has implications for which species are able to withstand not only the lower levels, but the higher fluctuations, right? Do species need to move in and out of this area in order to maintain oxygen levels that are acceptable to them? When we looked at the acoustic data, it was immediately apparent that we were looking at two very different kind of soundscape conditions. So of those 5 sites we recorded at, I'm only showing you 2 LTSAs here. The top one is for one of the devices that was in the submerged aquatic vegetation bed. The bottom one is for the water chestnut. All 4 devices that were outside of the water chestnut looked just like this LTSA. They looked pretty much identical, and it was only the water chestnut device which looked like this, which had much, much lower sound amplitudes across the whole analysis frequency band and through time. Now, first question, which might be occurring to some of you, is, was the device that was recording in the water chestnut actually functioning properly? Did it have a similar sensitivity to the other devices, or was it perhaps a low sensitivity, not reliable device? And so I went and did some playback experiments to these devices in very controlled settings in a lab environment, and found that the device that was recording in the water chestnut has comparable sensitivity to the other 4 devices. And so the soundscape differences that are seen here are real differences in the environment. They're not arising from an artifact of the device. After establishing that the data are reliable, our first question was, are these differences coming from differences in propagation or differences in acoustic community? By looking at this, you can already see largely that a lot of this is propagation, because most of these high amplitude noise events that you see here are the passages of vessels going by these sites. They are not biologic. And you can see that those vessel passages, while very low amplitude, they are somewhat visible at the water chestnut site as well, but much lower amplitude. So we went ahead and took well, we directly compared the amplitude of 10 high amplitude outside of the water chestnut vessel passages across sites, and we found that the amplitude that they were received at the 4 non-chestnut sites were all practically identical. The amplitude that they were received at within the water chestnut was much lower, which means that the propagation just through the chestnut itself is attenuating sound quite effectively, quite substantially. This isn't really surprising because the physical structure of the water chestnut is that there is a floating rosette of leaves and then a root filament that extends from that floating rosette all the way down to the sediment. And the root filament has leaves along its length as well. So you've got this very, very dense growth of thousands and thousands of surface rosettes, each attached to the sediment by a root filament and feathered with leaves along the lengths of them. So it's really a dense network, a mesh kind of plant matter throughout the water column. But we suspected that some of this difference in soundscapes might also be attributable to differences in community. And so that's what we're currently working up right now. And what we're hoping is that these insights can be useful to our Herner reserve managers as they plan for things like water chestnut removal in the future, to understand the acoustic community before, during, and after water chestnut removal, and whether or not water chestnut removal is able to restore kind of the more native community that we would expect in this area. Oh, sorry, that's just showing broadband sound level. So just the amplitude of sound averaged across our analysis frequency band through time. So it's the same as this, just summed up across frequency. The other follow-on project we wanted to touch on comes back to these freshwater drum again. So we mentioned that the freshwater drum chorusing was a really dominant component of the soundscape at the Atlantic sturgeon spawning site, which was surprising and news to us, news to our DEC collaborators. And so the question that immediately came out of that is there competition between these species for spawning grounds, for other resources, et cetera? And so we decided to use a sound mapping approach to better understand the kind of spatial overlap between these two species at this site. We went ahead and deployed a dense array of recorders. So we got low-cost, off-the-shelf recorders, and we put 25 of them out in this array, which is about 1/2 kilometer by 1/2 kilometer on each side. So the devices are separated by about 125 meters from one another. And we recorded just for a short time, just for about a week. Most of these low-cost, off-the-shelf devices have very, very short recording durations. And then what we're still in the process of, but I can show you some preliminary results, what we're in the process of right now is quantifying both freshwater drum chorusing and Atlantic sturgeon thundering at each of these 25 devices. Here I'll show you let's see if it'll play. This little video here is going to show you the intensity of freshwater drum chorusing at each of the devices over the course of just about a day. So this is the intensity of sound in the 200 to 400 hertz band. You can see these little quick flashes of high amplitude. That's when vessels pass through. And now you can see that the west side of the array is heating up. The east side to a lesser extent, mostly the west side. And now it's diminishing again. So that was the freshwater drum chorus for that day. And the video is going to loop so you can see it a few times. But what this is showing us already, this is just one day looping over and over. What this is showing us is that there is definitely a spatial pattern to the freshwater drum presence at this site. They seem to be favoring the west side of the array, which corresponds to deeper bathymetries and a different sediment type or a different substrate type than the east side of the array, which corresponds to shallower waters and rockier substrates. So next steps will be finalizing our sturgeon thunder time series at each site and making a similar visualization, and then quantifying the spatial overlap between these two species. So stay tuned for that. That's all that we have time to go over in terms of our kind of different analyses that we've explored so far for this project. I'm going to hand it back over to Maya to talk about some of the outputs from the project and our next steps.
Thanks, Rebecca. Yeah, so just to go down a list of some of the kind of key outputs that we were able to produce and then share throughout the course of this project. We presented a poster at the NERRS symposium, and then also a presentation which is available still online at the last NERRS annual meeting in 2023. We also had talks, one at the New York chapter of the American Fisheries Society, and then a talk at the North American Sturgeon and Paddlefish Society meeting. Rebecca also mentioned the soon to be, released paper kind of showing the sturgeon thunder, this first documented sturgeon recordings in the Hudson River Estuary. We also created a really interesting passive acoustic monitoring management guide. This is for fisheries managers, so non-bioacousticians, which is a tool to figure out, like, is acoustics for me? How can I use it? And then what is the step-by-step process for non-acousticians to utilize this technological and analytical process. And then we also wanted to share some of the educational outputs that we have created in order to reach a much wider audience with some of the sound analysis and recordings that were kind of created through this project. So here is one of the posters that we used throughout the project. It's posted up at the Norrie Point Environmental Center, as well as on the NERRS Science Collaborative project page. One of the things I really like about this particular poster is that you can see the QR codes that are embedded in the poster all have sound clips, so you can actually hear the thundering of the sturgeon when you're visiting the poster, or you can hear some of the soundscapes from different sites. So this is one of the outputs that we wanted to share. Another is just so we have a lot of students that come to the research reserve on the Hudson, and we were able to pilot and then put into use many different interactive acoustic activities, including mapping soundscapes. So students are out in the estuary watershed, drawing the different sounds that they're hearing, as well as trying to identify whether it's birds or fish or amphibian sounds that they're able to hear in real time. We also created what we are calling the HELLO, which is the Hudson Ecological Listening Laboratory and Observatory. It's a little sound booth that's at the Norrie Point Environmental Center using Cornell Lab of Ornithology's Raven exhibit software. You can see students can come into this little kind of sound booth and then choose different animals that they would like to listen to. And there are clips from around the world, but we also highlighted some of these, like the Atlantic sturgeon, and then some of the geophonic sounds, so like the sound of a storm passing through one of the reserve component sites. And then we also created a couple of different story maps. One is it's a voyage up the Hudson River while listening to the sounds in the different estuary sites, whether it's urban environments or very protected wetland environments. And then an accompanying lesson plan for students to use during classroom hours or as homework lessons to kind of extend the learning beyond being at the actual river's edge. We also had quite a few students that were interested in conducting research of their own or helping us with our research. So we developed a deeper listening story map that is actually like preparation or training for students interested in doing bioacoustics. So it's more of an acoustics 101 in the Hudson River and how to use this technology in some of the analytical processes. Also, just the participation of so many different students at all different levels was one of the like really shining highlights of the educational outputs. We had students that did posters, students that helped collect data, students that did their own mini projects from high school, college, graduate, all different levels that participated in collecting sounds and trying to understand the eco soundscapes. So we learned a lot. We just wanted to highlight a couple of things. One was that this was, through science collaboration, we were working with a bunch of different agencies that all have different goals and methods and skills. And so coming together with all these different agencies, whether it's HERNER, the education and research sectors, the New York State Department of Environmental Conservation, the Fisheries Unit, the Management Program, all these different entities coming together was both challenging but also very satisfying to bring everyone together. Then also one of the things that we learned a lot about was different entities and their like field seasons or their teaching seasons, and everyone had a very cyclical annual schedule, and we had to adjust quite a bit of our expectations for our end users and stakeholders based on the other kind of seasonal needs that went with their jobs. And then also, as Rebecca was showing you the different methods that we use, we also had very different equipment, and some of the described as like cheaper off-the-shelf equipment, was very limited, whereas some of the, investing in some of the more expensive sound traps that could give us much longer recordings, but were also much more expensive, did give us a lot better data. So that was a lesson learned, is that if you're doing acoustics, invest in the more expensive or better recording equipment. We have a lot of future directions. Science Collaborative project has really just sparked quite a bit of other future work for us, including continued analysis of the data that we have already collected. We have, tons and tons of data that still is waiting for further analysis. We're also now working with the invasive species team in order to evaluate some of this water chestnut removal potential, to see how management, can be informed by the acoustic data. We also are continuing to work with the fisheries unit at the DEC, as the mapping of the habitat with drum and also with the Atlantic sturgeon proved to be very valuable for them. And then we're also looking at follow-on projects using what we've learned about Atlantic sturgeon and short-nosed sturgeon in the Hudson, and extending to other ranges of their habitat, including much further north in the Kennebec River. So we wanted to acknowledge, contributions from so many people, folks at the Norrie Point Environmental Center, students, including students from West Point Academy and Bard College, Marist, SUNY New Paltz, Cornell, many high school students. We also had interns from the Student Conservation Association help, and lots of input from Hudson Valley teachers and educators in designing, the story maps and the lesson plans. And then a special thank you to our funders, NOAA, NERRS, and Science Collaborative, for supporting all this valuable work, and especially Jen Reed for her ongoing support as this project rolled out. And then to Fatima and Alexandra for infographic development. And I also wanted to thank all of you who have been on the call. At one point we hit 106, and so I want to make sure we leave a little bit of time for 106 questions in case everybody has something that they wanted to say. So at this point, I'll open it up to questions, and I'm going to leave our contact information up here in case anybody wanted to do a screen grab and reach out to us afterwards, in case we don't, for some reason, have enough time to answer everyone's questions. So thank you all so very much.
Great work, folks. Thanks so much. We do have a few questions here I see in both the chat and the Q&A. So I'm going to copy them over, and I will put them into the Q&A so that everyone can see the ones that are in the chat too, because that happens sometimes. If you do have questions, just feel free to pop them in. So let's see here. The first question I see, other than the known spawning or overwintering aggregation areas, are any of the other main channel sound traps associated with specific benthic habitat types?
Sorry, couldn't unmute myself. Most of the Hudson is quite a muddy, silty bottom, lots of organic matter being swept down the river. So it's really only the Atlantic sturgeon spawning site that we were monitoring in kind of a rocky, gravelly area. And that's actually thought to be why the sturgeon are spawning in that specific location, because the Atlantic sturgeon eggs at least need a gravel bottom. They adhere to the rocks, and if they are deposited in a soft, silty bottom, they get coated and covered and the eggs suffocate. But no, the other sites were all pretty soft bottom, as evinced by the muck that we often would pull up when pulling our devices back out of the water. And they were not, the other sites were not chosen to specifically target particular species or behaviors in the same way that the Atlantic sturgeon spawning site was. That one was really unique in that regard.
Okay, got a few minutes left here, so we'll see how many we can get through. Somebody had posted a question in the Q&A, so I'll post it in the chat just so everybody can see it. Oops, that's just to the host and panelists, though. Might help if I gave it to everybody. Is it both male and female sturgeons that vocalize, or just one sex or the other?
So that's not definitive yet for the Atlantic sturgeon, but
Oh, interesting.
our best guess for the mechanism is when the female sturgeon are releasing their eggs, the males come along up alongside them and they kind of do this full body thrashing as they're releasing their own sperm to fertilize the eggs. In the lake sturgeon, the the kind of timpani beats in that thunder roll that they produce seems to correspond directly to the thrashes of the males as they release their sperm. So it seems like their full body muscular contractions may be causing the swim bladder to reverberate. So if that is in fact the mechanism for sound production, as we think it is, then it's just males who are producing these thundery sounds.
Interesting. Okay. Another question came in. Just curious what software is used to calculate the soundscape metrics. And I think we talked about that beforehand, but this is a cool answer, so go ahead.
Well, it's actually a little different than what we were talking about, Nick. So
Okay.
for the soundscape metrics, I used MAMTA, which is a noise analysis software that takes raw files, preferably from calibrated devices such as the sound traps, and calculates absolute receive level. So decibels relative to whatever your reference pressure is, 1 micropascal for underwater recordings. Um, yeah, that's what I used here. I won't go into more detail there, but if you want to talk more detail about analyses, I'm happy to do that and reach out to me afterwards.
Okay. Next question. Have you considered incorporating eDNA analyses into the project?
That's not something that we within our group have undertaken, but our collaborators at New York DEC are in fact doing a bunch of eDNA. So Rich Pendleton there has been heading up eDNA for at least the last couple of years, and he's been focusing on a number of species, including a bunch of stuff on short-nosed sturgeon and maybe to a lesser extent Atlantic sturgeon, but looking at things like uh, the population origin of the individuals that are, well, for the Atlantic sturgeon, population origin of the individuals that come back into the river to spawn. Some of that's, oh no, I'm sorry, that's actually from the biopsy stuff. Maya, you might be able to speak to this better than me, actually.
Yeah, so from my understanding, a lot of the eDNA work that Rich Pendleton from the fisheries unit with the DEC has been doing is they're looking at some of the like new invasives coming down. So they're looking at things like round goby, which actually is thought to be a vocalizing fish. In some ways, like using bioacoustics and eDNA in concert with each other are very effective, and in some ways using eDNA to see the emergence of a new species is more effective, and then with a bioacoustics recording where there's known already populations rather than looking for like the very few, So yes, we are involved with partnerships that are using both technologies. But at least within the team that's presenting today, we're more of the passive acoustic knowledge.
Nick, do you mind if I jump in? I noticed a
Yeah.
question from Jules Jaffe in the chat. Hi, Jules. Nice to see you. There are a lot of tagging efforts going on for the sturgeon, actually, for both the Atlantic sturgeon and for the short-nosed sturgeon. So the correlation between the occurrence of sturgeon, Atlantic sturgeon thunder and the presence of fish was based on telemetry tagged fish. So yeah, that's been a really powerful tool for knowing when the animals are in the system, where in the river they're going, et cetera. And actually based on that, um, the Hyde Park spawning site has been known for a long time. There's also historic evidence that suggests that there might be a secondary spawning location for Atlantic sturgeon, which is a little further north in Catskill. And a very small proportion of the tagged fish do proceed all the way up to Catskill each year. And so what we hope is that perhaps next year we could throw a device out there and do some recordings up there and determine if in fact there are individuals spawning up there, because that area is not currently being studied or protected in the same ways as the Hyde Park spawning site. So that would be a neat finding.
For the folks just listening along, that was in response to the question, have you thought about tagging, fish tagging to track the sturgeon? Okay, we have actually about 1 minute left, so it might be good to just call it there. I think there were a couple questions that were unanswered, but I will pass those along to presenters and we'll make sure to include them and maybe we can have them follow up via email. Thank you to everyone for joining us today. If you look out for the recording, you can usually find it done within 1 or 2 days. It might take us a bit longer this time, just given the audio files we need to make sure will come through in the recording. So it might take a little bit longer, but hopefully still within a few days. You can check back to the Science Collaborative's website or our YouTube channel. And if you signed up for the webinar, you should automatically get an email that tomorrow, same time, that has the link to the summary resources, and you should see it automatically. So, um, thank you again, everybody, for joining us. Thanks so much to our presenters for putting together such a great presentation. And this is a really fun one. So, everyone, have a great rest of your day.
Thank you.
Thanks a lot.